Study Awesome Tutor, and today we're going to be looking at enzymes. Now, enzymes are biological catalysts. What is a catalyst? Well, a catalyst speeds up the chemical reactions which take place, in this case, because they're biological catalysts, inside your cells. But catalysts, whilst they are catalyzing the reactions, they are not used up themselves. So they can be reused. And different enzymes have different shapes and therefore different functions, because the sequence and type of amino acids in their structure is different. Okay, enzymes are made up of amino acids. And what is an amino acid? Well, amino acids make up protein. So an enzyme is basically a protein. Okay, and the sequence of amino acids in its structure, they determine the specific shape, because enzymes are specific. The long chains of amino acids, they fold to form a molecule with a specific shape. And this molecule with a specific shape is the enzyme. Next. Okay, so I'm going to keep stressing this. Enzymes are specific. They are specific about what reactions they catalyze. Why are they so specific? Because their shape is specific. Okay? The long chains of amino acids, they fold to form a molecule with a specific shape. And this specific shape is really important. Why? Well, let's see. Here we have the enzyme. Here we have some molecules. Which molecule do you think will fit into the enzyme? Obviously, it's going to be this one. And this is what we call the reactant. The reactant molecule. Or sometimes it is referred to as the substrate. Reactant or substrate. And then the enzyme and the reactant combine, or the substrate, to form an enzyme-reactant complex. And in the case of digestion, the enzyme breaks down the molecule, and then, of course, the products are released. Here we have the products. Okay, so here, the place where the substrate and the enzyme will bind is called the active site. And the active site is obviously going to be a specific shape, because that's where the substrate will fit onto it. If the shape of the active site is different, the substrate cannot fit onto the enzyme. And so the active site has to be a specific shape to match the substrate. Yes, next. Okay, so what happens if we increase the temperature? Well, from chemistry, rate of reactions, if we increase the temperature, usually the rate of reaction will increase. And yes, if you increase the temperature, the rate of reaction will also increase. But then what happens here? Well, your body must be kept at a constant temperature, 37 degrees Celsius, because this is the temperature that your enzymes work best at. If the temperature drops a few degrees below 37 degrees Celsius or a few degrees above, the enzymes will not be able to work. They will not be able to function, and because they control the rate of reactions, remember they are the catalysts, the rate of reaction will go down. So if the temperature is too high, we say the enzymes denature. And denature means that they stop functioning. Okay, here we have an enzyme at a temperature of 37 degrees Celsius. Okay, normal shape, it can fit with the substrate. Here we have an enzyme at 60 degrees Celsius. You can see that its shape has changed. So if the temperature is too high, the shape of the enzyme changes. And because the enzyme's shape is very important, because it's specific to what molecule it will fit onto, if the shape changes, it can't fit onto this molecule. anymore. The substrate in the enzyme cannot bind anymore. And so they stop working. The enzymes are now useless because if they can't bind to the substrate, they can't catalyze the reaction. Okay, and this happens because the chains of amino acids, they begin to unravel because of the high temperature. And also, different enzymes work best at different pHs. But we're going to look at that more when I talk about enzymes in digestion in a later video. This has been the Awesome Tutor. Bye!